Okay, hello everyone. Hello everyone, this is Dr. Priyankita and I am going to start the rapid revision of anatomy, neuroanatomy part 3. We have already discussed the neuroanatomy part 1 and part 2. Most of the topics has been covered. So whatever is left behind, we are talking on this session. Okay, so let us start. Okay, first question is notochord is formed by. So they have given nucleus pulposus, hypoblast, epiblast, and myeloblast. So anyone who knows this answer in the chat, if anybody wants to answer this, किसको मालूम है ये answer? So before starting this, let us study a little bit about the embryology. Okay, if we study the embryology, we'll get to we'll answer this question properly. See, let us see the formation of blastocyst. Okay, you know that there are blastomeres, right? 2-cell stage. You have a 2-cell stage. Okay. Then you have 4-cell stage, then 8-cell stage, and 16-cell stage. That is the morula. Okay. Uske baad there will be a formation of the early blastocyst and the late blastocyst. So some cells will go on the side. Okay. Some cell will go on the side, and few cells will accumulate on one side. ठीक है. इतना तक समझ में आ गया. And there is a first cavity form. Okay, cavity 1 form. Now this cell which has gone on one side, one end, is known as the inner cell mass, and the cells which are outside, okay, that outer cell mass is the trophoblast. ठीक है. So इतना तक समझ में आ गया कि first we have the blastomere 2-cell stage, then 4-cell, then 8-cell, then morula. Then after this cleavage, we got that we have an inner cell mass and we have a cavity. ठीक है. The inner cell mass is going to one end and the cavity 1 is formed, and then you have the outer cell mass, that is the trophoblast. Okay. Now what happens? See, this is known as the cavity of the blastocyst or the blastocele. Okay, and this part is known as the polar trophoblast, the one which is near the inner cell mass. Okay, and the outer trophoblast is known as the mural trophoblast. So itna tak to you know only. After that, what happens? Okay, after that, what happens? So you know till this much, you know that this is the inner cell mass and this is the outer cell mass, that is the trophoblast. Okay, now there will be formation of the bilaminar germ layer. Okay. So in the week 2, you know that in the week 2 there is rule of 2. So you have the formation of the bilaminar germ layer. So this outer cells, upper cells, okay, they are dividing into 2 layers. This will be columnar and they are known as the epiblast. ठीक है. और जो नीचे का cell है, it will be little bit cubical. Okay, cubical or cuboidal, and those are the hypoblast. ठीक है. So this inner cell mass is again dividing into your epiblast and the hypoblast. Okay. Now there is another cavity developed in this epiblast, that is known as the amniotic cavity. Okay. This is the cavity 2. This is the cavity 2. Okay, and we already have a cavity 1 here which is forming the primary yolk sac. Okay, the blastocele cavity is forming the primary yolk sac. Okay. Okay, the blastocele cavity is forming the primary yolk sac. Okay, and then we have the cavity 2, that is the amniotic cavity. Now we have 2 cavities. Okay, and this is again the epiblast and this is the hypoblast. ठीक है. उसके बाद क्या हुआ? Can you see certain cells of this hypoblast are migrating? Okay, are migrating on the sides. Okay, and then they are forming the extra embryonic membrane. The extra embryonic membrane. Okay, after that, from the epiblast, you have the formation of the 3 layer, that is the ectoderm, mesoderm, and endoderm. So after the bilaminar germ disc, you will have the formation of the trilaminar germ disc. Okay, and there you can see that there is a formation of a rod-like structure from the epiblast, and that is known as the primitive streak. Okay, that is known as the primitive streak. So till this much you can understand, I guess. Any doubt? So पहले क्या था? You have this entire structure where you have the inner cell mass and the outer cell mass. The inner cell mass is again dividing into the bilaminar germ disc, that is epiblast and hypoblast, and you have a blastocyst cavity. Okay, this is cavity 1. After that you have the formation of the cavity 2, that is the amniotic cavity, and cavity 1 is forming the primary yolk sac. You can also see the hypoblast cells migrating towards this primary yolk sac to form the extraembryonic membrane. Okay, then from the epiblast you have a formation of rod-like structure, that is the primitive streak, and then this bilaminar will ultimately divide to form the trilaminar, that is ectoderm, mesoderm and endoderm. So it's clear. Okay, so gastrulation, the gastrulation is in the week 3, and it is the conversion of your epiblast from the bilaminar into a trilaminar embryonic disc. So gastrulation is the from bilaminar to trilaminar, that is gastrulation. And then you will have all the 3 parts, that is ectoderm, mesoderm and endoderm. Now gastrulation begins with the formation of the primitive streak, and this occurs at the 3rd week. Okay, 3rd week. The gastrulation process occurs at the 3rd week. Okay, the bilaminar, the rule of 2, 2nd week you have the bilaminar disc, and in the 3rd week you have the trilaminar germ disc. Okay, okay, that is about embryology. Now what happens? We are going through the step by step so that we can answer the notochord. Okay, so for that you have to also understand the embryogenesis a little bit, otherwise you will not get the point. कहाँ से notochord आ रहा है, just mugging up will not help you. Okay, so now you can see that in this image you can see this is the amniotic cavity. Okay, this is the amniotic cavity, and then you have the embryonic disc. Okay, and this is the primary yolk sac. Also you have the extra embryonic membrane. Okay, after that you can see the germ disc, amniotic cavity. There is this amnion and the chorion formation. You have another part, secondary yolk sac, and then you have the extra embryonic coelom. Okay, so till this much it is clear, and the trophoblast is again dividing into your, one is the splanchnopeuric mesoderm. Okay, the splanchnopeuric mesoderm. This is the splanchnopeuric mesoderm. This is the splanchnopeuric mesoderm, and the outer part is known as the somatopleuric. Okay, the trophoblast is again dividing into the splanchnopeuric extraembryonic mesoderm, and outer it is soma. Soma means body. So outer part is the somatopleuric extraembryonic mesoderm. ठीक है, तो ये हो गया. See, in this image also it is clear that you can see the rod-like structure here, that is the primitive streak. Okay, you can see the rod-like structure, that is primitive streak, and then you have formation of ectoderm, mesoderm and endoderm, that is in the week 3. Okay, then you have the yolk sac, that is the secondary yolk sac, the amniotic cavity and the extraembryonic coelom. Okay, this outer part is the somatopleuric. Okay, somatopleuric, and the inner part is the splanchnopeuric. Okay, so you have the splanchnopeuric mesoderm and a somatopleuric mesoderm. So that's about the embryogenesis. Then what happens? Now we have to understand the formation of the notochord. Now notochord formation will undergo the following processes. Okay, first of all there is the formation of the precaudal plate. Okay, precaudal plate. After the formation of precaudal plate, you will have the formation of the notochordal process. Okay, there is formation of the notochordal process. Then there will be notochordal canal formation. After canalization you will have neuroenteric canal formation. Then you will have notochordal plate, and then you will have the definitive notochord finally. Okay, so now I want to say that you can see this image, right? You can see this image. Now if we cut from here, and remove the amniotic sac. Okay, remove the amniotic cavity and see from above. Okay, if we cut this amniotic, this entire part, and we see this entire structure from above, then you will see this embryonic disc. First it is round and then it becomes elongated. Okay, now in the embryonic disc you can see, first of all, there was what you have, the columnar. This is the epiblast, and now this is the epiblast, and then you have the cubicle below, that is the hypoblast. So you're seeing that embryonic disc from above, so you can see the columnar ectoderm and the cubicle endoderm or the hypoblast. Okay, now certain cells, now you have two ends here. So one is the head end, that is the cranial end. This is the cranial end. Okay, and the other one is the caudal end. Okay, the caudal end, the cranial end and the caudal end. Now towards the cranial end, some endodermal cells, okay, some endodermal cells becomes columnar. Okay, the endodermal cells, that is from the epiblast only, they become columnar, and this results in the formation of the precaudal plate. Okay, precaudal plate. And then towards the tail end, towards the tail end, towards the tail end, there will be formation of the primitive streak. So पहले हो गया तुम्हारा precaudal plate formation in the head end. Okay, so now you have understood the orientation. So पहले बन गया your precaudal plate. You have the precaudal plate in the head end. Okay, this is in the head end or the cranial end, and then you have a caudal end here. Okay, so the primitive streak, the ectodermal cells will proliferate here. In this area, the ectodermal cell will proliferate here, and this will result in a rod-like structure that is known as the primitive streak. Okay, that is known as the primitive streak. So you can see here the ectodermal cells are proliferating, and that is resulting in the primitive streak. Okay. So you can see that the primitive streak is from the epiblast only. Okay, the primitive streak is a linear band of thickened epiblast, and it appears in the caudal end first, and then it will grow towards the cranial end. And this cell will ultimately proliferate towards the cranial end to form a primitive knot. Okay, a primitive knot, and this primitive knot is also known as primitive node or Hansen's knot. Okay, so what happens? You have this embryonic disc, okay, and then you have the precaudal plate, caudal plate. Okay, this is the head end and this is the tail end. Okay, now there is development of the primitive streak. Okay, there is development of this rod-like structure. There is development of this rod-like structure that is known as the primitive streak. Okay, which will grow towards the cranial end. Okay, it will grow towards the cranial end, and then okay, so there is a formation of this rod-like structure. Okay, it is going towards the cranial end. It is going towards the cranial end, and then what happens? It is forming a node. Okay, it is forming a node, and you will also have a pit here. Okay, so this green color is the primitive pit, and the outer part is known as the primitive knot or Hansen's knot. Okay, so till this much you understood that there is the precaudal plate and there is the primitive, this is the primitive streak. Okay, so precaudal plate is in the head end and the primitive streak is in the tail end. So first there is primitive streak, it is going towards the cranial end, and then it is forming a knot that is known as the primitive knot or the Hansen's knot, and then you also have a pit there that is known as the primitive pit. Okay, so till this much it is cleared. Okay, so what happens then? The mesenchymal cells from the knot, primitive knot, will form a midline cellular structure that is known as the notochordal process. So we till this much you know that this is the precaudal plate. Okay, this is the precaudal plate. Okay, this is the head end, and then you have the primitive streak. Okay, the primitive streak will develop. Okay, the primitive streak will develop a primitive groove. It's going to develop a primitive groove. Okay, the primitive streak is going to develop a primitive groove. Then you also have the primitive node or primitive knot, and then you have a primitive pit. Okay, you have a primitive pit. Now from this primitive node or primitive Hansen's node, certain mesenchymal cells will proliferate, and they are going to form again a rod like substance that is known as the notochordal process. Okay, it is known as the notochordal process. So that is what they mean by notochordal process. Okay, now this process is going to grow towards the precaudal plate, which is going to finally form the future mouth. Okay, and also it is going to grow towards the tail. Okay, it is also going towards the primitive streak to form the cloacal membrane, that is the proctoderm or the primordial anus. Okay, so this process will grow towards the cranial end to form the mouth and go towards the caudal end to form the anus or the proctoderm. Okay, so that is about a notochordal process. Now what happened? Now this process, which is a notochordal process, certain cells will degenerate. Okay, certain cells will degenerate, and they are going to form, they are going to invade on themselves. You can see that the amnion will invade on the primitive streak in the notochord, and they are going to canalize. Okay, so what happens? This notochordal process, which is a solid rod like, that will undergo a canalization. So that will undergo a canalization. Okay, you will have a canal in between form, and that is known as the notochordal canal. So you can see here in this image as well. So you have a primitive pit or the blastophore, and then you got the notochordal process. Okay, you got the notochordal process here. Okay, and then how the cells invaginate, and then you have the formation of a canal like structure, and that canal is known as a notochordal canal. Okay, now after the notochordal canal, there will be another canal that is known as the neuroenteric canal. Okay, that is known as the neuroenteric canal, and that happens when the floor of the notochordal canal degenerates. Okay, and the remaining roof will form the notochordal plate. Okay, and this will ultimately proliferate and detach from the endoderm, and then this plate is going to form the definitive notochord, which will underlie the neural tube. Okay, so these are the processes. So what we understood that from this image, it is very easy to understand that पहले क्या था. We are seeing the embryonic disc, है ना? You know that we have removed the amniotic cavity. We have cut in a horizontal plane, and we are looking from up, so you can see a precaudal plate formation, which is towards the head end. First there is a formation of precaudal plate, which is towards the head, and then you have a formation of a streak, a rod. You have a formation of a rod, that is the primitive streak in the caudal end. Then what happens? This primitive streak is going to form primitive node. Okay, primitive node, and from the primitive node, certain cells are going to come in the midline, and they are forming the notochordal process. Now notochordal process first is it is solid rod, then it is going to form the notochordal canal, then neuroenteric canal, and then there will be the formation of the notochordal plate. Okay, and this notochordal plate is ultimately, this notochordal plate is ultimately going to give rise to the definitive notochord. Okay, so it is going to give rise to the definitive notochord. Okay, so that is how the notochord is formed. So similarly here you can see that there is a notochordal process, then notochordal canal. Okay, then you have the neuroanteric canal. After that you will have the notochordal plate. Okay, and then you will have the definitive notochord. We are going to have the definitive notochord. Okay, so that is how the notochord is formed. Okay, then there will be the formation of the neural tube. Okay, now how the neural tube is formed. Before going to the neural tube, let us see the fate of the notochord, that from where it is arising. Okay, so you can see that you have the blastocyst first. Okay, pehle blastocyst. So now it is going to divide into the inner cell mass and the trophoblast. Okay, the blastocyst is dividing into the inner cell mass and the trophoblast. Now trophoblast is dividing again into the syncytiotrophoblast and cytotrophoblast, outer layer, and the inner syncytiotrophoblast. Okay, till this much it is clear. Let us go to the inner cell mass. Now you know that in the week 2 you have the epiblast. You have the epiblast and you have the hypoblast. Epiblast is above, the hypoblast is below. Okay, now from the hypoblast certain cells are migrating downwards towards the primary yolk sac, and they are forming the extra embryonic endoderm. Okay, and that is giving rise to the yolk sac. Okay, extra embryonic endoderm, and they are forming the yolk sac. That is from the hypoblast. So hypoblast is giving rise to your extra embryonic endoderm. Okay, extra embryonic endoderm. Now from the epiblast again you have two types. One is the amniotic ectoderm. One is the amniotic ectoderm, the ectoderm towards the amniotic cavity, and one is the embryonic epiblast. Okay, amniotic it is epiblast is giving rise to amniotic ectoderm, and then it is giving also have the embryonic epiblast. It is also known as the embryoblast, this embryonic epiblast. Now this embryonic epiblast is giving rise to all the three layers, that is the ectoderm, endoderm, and mesoderm. So you have from the embryonic epiblast, it gives rise to the embryonic ectoderm, and from this embryonic ectoderm you have the neural crest cells. Neuroectoderm. Okay, this embryonic ectoderm is also the neuroectoderm. So you are getting the neural crest cells from the embryonic ectoderm. Again, we know that the primitive streak, okay, from the embryonic epiblast, there is a formation of the precaudal plate and the primitive streak. Now, the primitive streak, first of all, the primitive streak will give rise to your embryonic endoderm, okay, embryonic endoderm and the allantoic endoderm, okay, and again it is divisible into three parts: extra embryonic mesoderm, embryonic mesoderm, and the notochordal process, which is also a mesoderm in origin. Okay, so from the primitive streak the notochord is arising. So this is the entire chart. Okay, if you have to understand, again I am repeating that pehle tha blastocyst. Okay, then you have the blastocell cavity, you have a inner cell mass and a outer cell mass. The outer cell mass is known as a trophoblast, which is giving rise to your cytotrophoblast and syncytiotrophoblast. Okay, inner cell mass at the second week it is seen as two layer bilaminar germ disc. So you will have the outer epiblast and below you have the hypoblast. Hypoblast cells are migrating towards the yolk sac, okay, and that is forming the extra embryonic endoderm. Okay, the extra embryonic endoderm, and then you have the yolk sac formation. So yolk sac is ultimately coming from the hypoblast, okay, hypoblast, and then from the epiblast you have again amniotic ectoderm and embryonic epiblast. The epiblast is such a layer that it is giving rise to all your the ectoderm, endoderm, and mesoderm. So first of all you have the amniotic, the one amniotic cavity lying the amniotic cavity. So you have the amniotic ectoderm, and then you have the embryonic epiblast, which is also known as the embryoblast. Now embryonic epiblast is again divided into the embryonic ectoderm. Okay, I told you from the embryonic epiblast you are going to get all the three layers. So you have the embryonic ectoderm, which is also known as a neuroectoderm, and it is giving rise to your neural crest cells. And then you have the primitive streak. Now primitive streak is giving rise to your extra embryonic mesoderm, the embryonic mesoderm, and the notochordal process. And also it is also giving rise to your endoderm, embryonic endoderm and allantoic endoderm. Okay, so this is the entire hierarchy. So you can see that notochord is ultimately arising from your primitive streak, and primitive streak is again arising from your epiblast. Okay, so ultimately notochord is coming from the epiblast. Okay, it is coming from the primitive streak, which is nothing but formed from the thickened epiblast. The primitive streak is formed from the thickened epiblast. Both the precaudal plate and the primitive streak is from the epiblast. So the answer to the question, is we have given that notochord is formed by nucleus pulposus, hypoblast, epiblast and myeloblast. So the answer is your epiblast. Okay, now one more thing I want to tell you, that remnant of notochord is the nucleus pulposus. You know that there is an intervertebral disc, and in the center of the intervertebral disc you have the remnant of the notochord that is known as the nucleus pulposus. Okay, and then you also have a ligament that is known as the apical ligament of Dens. Okay, so these are the two remnants of notochord. Okay, so notochord is not formed from the nucleus pulposus. Nucleus pulposus is the remnant of the notochord, and you also have the apical ligament of Dens. Okay, there are two remnant of notochord, the nucleus pulposus in the intervertebral disc and the apical ligament of Dens. ठीक है? Okay, now till this much we know that notochord is formed, neural plate is formed, notochord is formed. Okay, now simultaneously what happens? Okay, simultaneously you can see here, you can see in this image first. Let us see this image. Now we want to see how the neural tube is formed. तो हमने पहले notochord पढ़ लिया है, how the notochord is formed. Now we are going to the neural tube. Any doubt? You can ask in the chat. Okay. Okay. So here you can see that there is this ectoderm. This is the ectoderm. Okay. And then you have the endoderm. All are arising from the epiblast, and then you have the mesoderm. Okay, mesoderm, and this is the notochord. Okay, this is the notochord. This is the notochord. Now you know that from the ectoderm, that is from the epiblast, you have that neural plate. Okay, so this is the neural plate formation when the notochord was formed. Okay, neural plate formation, and this is the neural plate. And what happens? This neural plate will invaginate down. Okay, this neural plate will invaginate down, and then a groove is developed, and this groove is your neural groove, and on the sides you have certain cells, that is this, not cells, this side area is known as the neural crest, because you have a crest and then you have a groove. When there is a thing like that, when you have a thing like that, you know this is the crest, this is the crest, and this is the groove. This is a groove. Okay, so the side part is the crest and that is the groove. Okay, and simultaneously you can also see here that there is that intra-embryonic coelom developed. Okay, intraembryonic coelom develops. So you have the intraembryonic mesoderm. Then what happens after it goes down? This invaginates down and it forms a tube. Okay, it forms a tube. This groove forms a tube, and that tube is known as the neural tube. And on the sides crest will also disintegrate to form the neural crest cells. Okay, and you know that the notochord lies below the neural tube. Okay, and then you have the intraembryonic coelom. Okay, you have the somite formation also. Okay, so same thing if you see here in this image, this is the न्यूरल प्लेट. ठीक है, ये है न्यूरल प्लेट, ये एक्टोडर्म है, दिस इज द मेसोडर्म, एंड देन दिस इज द notochord which is already formed. We have already studied. Now how there is the convergence, okay, how the neural plate is invaginating and forming the neural groove, and then you have this area that is the neural crest. Okay, neural groove and the neural crest, and after that this neural groove is finally forming the neural tube, and the neural crest cells are on the sides, which are the components of the PNS. Okay, and below you have the neural notochord. Okay, this is the neural tube and notochord. Okay, so finally you can see that this is the epidermis. Okay, this is the neural tube, and then you have the neural crest cells that is forming the spinal ganglion. Okay, and the notochord below, and these are the somites. Okay, that is how this entire neural tube is formed. So neural tube पहले क्या होता है? पहले होता है neural plate. Okay, first you have the neural plate, then it is going to invaginate. So invagination forms the neural groove, and the outer elevated area is the neural crest. Then this groove is transformed into the neural tube, and then the cell disintegrates to form the neural crest cells, part of the PNS. Okay. So now we have studied about the neural tube. Okay, so one more thing I want to explain, that let us see the neural tube, how it is formed. Now neural tube is open, okay, cranially and caudally. Okay, you can see here, this is the neural tube. Okay, and there is an opening in the anterior part, and there is an opening in the posterior part. Okay, so the anterior opening is known as the anterior neuropore, and the posterior opening is known as the posterior neuropore. And many questions is, if you're correct with the concept of the anterior neuropore and the posterior neuropore, then you can answer many questions. So now the question is that the anterior neuropore will it will get closed on 25th day of the fertilization, and the posterior neuropore will close on the 27th day of the fertilization. That is about the neural tube, the anterior neuropore and the posterior neuropore. Okay, so we will discuss the neural tube defects now. So you know that how the neural tube is formed. Okay. Now before going to the neural tube, let us little bit attempt the embryogenesis question. So hypoblasts form, they have given precaudal plate, notochord, apical ligament of dens, and endoderm. So out of this, anybody who can answer this question, hypoblasts will form which of the following structure? Anyone in the chat who wants to answer this, hypoblasts form which of the following structure? Just now we have seen a chart. Okay, in the chart we have seen that there is inner cell mass and there is the outer cell mass, the tropoblast. Inner cell mass is giving rise to epiblast and a hypoblast. Now I have already told you epiblast is giving rise to all the three layers. Now from the hypoblast, what are the structures formed? So you know that, try to पहले देखो कि क्या क्या नहीं होता है. So precaudal plate, you know that it is formed from epiblast. Notochord is also formed from epiblast. Apical ligament of dens is remnant of notochord. So what is the answer remaining? That is the endoderm. So we know that the extra embryonic endoderm is formed from the hypoblast. Okay, the extra embryonic endoderm is formed from the hypoblast. Okay. Now gastrulation occurs in which week? They have given first, second, third week. Anybody who wants to answer this? Gastrulation. Very easy. Gastrulation occurs in which week? Anyone who wants to answer this question? Gastrulation occurs in which week? You know that rule of 2 is bilaminar, and gastrulation means from the bilaminar to trilaminar disc. Okay, from the bilaminar to trilaminar disc, that process is known as the gastrulation, and that is in the week 3, third week. Okay, gastrulation occurs in the third week. Okay, so these are a few questions from the embryogenesis part. Now, there is a very important topic which is to be discussed, and this is repeated in the NEET PG. Also, you might also get a question, picture related question in the FMG as well. Okay, many times it is repeated in the NEET. So, they have asked in this question, which of the following statement is incorrectly related to the given image? So, they have given an image here. Okay, so in the region of the sacrum and the coccyx, you can see a very large tumor. Okay, it is a very large tumor, and this tumor, they have given what is the incorrect answer. So they have given it is a benign tumor of coccyx, presence of teeth and hair, results from aberrant migration of the primordial germ cell, and precaudal plate anomaly. So this condition is known as the sacrococcygeal teratoma. Okay, this is known as the sacrococcygeal teratoma. This condition is known as the sacrococcygeal teratoma, and this arises, since it is teratoma, these teratomas will arise from all the germ layers, that is the ectoderm, the mesoderm and the endoderm, and they are going to have your, since they are arising from all the germ layers, they are going to have your the teeth, the hair, okay, all the germ layers derivatives. Now they are believed to be the remnant of the primitive streak. You know, primitive streak is from the epiblast, right? They are believed to be the remnant of the primitive streak in the coccygeal region during the early week of gestation from the totipotent cells of the Hansen's node. Okay, so this is the definition of your sacrococcygeal teratoma. Now let us come to this question. They have mentioned that it is a benign tumor of the coccyx, yes. Presence of teeth and hair, yes. Results from aberrant migration of primordial germ cell, yes. So the answer is your precaudal plate anomaly. It is not a precaudal plate anomaly. Okay, precaudal plate anomaly will be towards the mouth. Okay, it will be not towards the sacrum or the coccyx. So it is not a precordial plate anomaly. Okay, so it is due to the remnant of the primitive streak. Okay, this is sacrococcygeal teratoma is due to the remnant sacrococcygeal teratoma. Okay, this is due to the remnant of your primitive streak. Okay, this is due to remnant of your primitive streak. So the answer here is obviously this is precaudal plate anomaly. Precaudal plate, you know, it is towards the head end, so it is going to give rise to the future mouth. Okay, so it is not the incorrect one is the precaudal plate anomaly. Okay. Now we got to know about the neural tube and the anterior neuropore and the posterior neuropore. Okay. Now, let us come to the neural tube defects. So, if the whole length of the neural tube is unclosed, okay, अगर पूरा length ही unclosed है, if the whole length of the neural tube is unclosed, the whole length, okay, of the neural tube is unclosed, then it is mostly going to give rise to a condition known as a posterior echichiasis. Okay, it is also known as a cranioechichiasis. So you can see in this image that the entire neural tube is open. The entire neural tube is open. So this is a condition of your posterior echichiasis or cranioechichiasis. Okay, every time asked in the exam, the echichiasis, the spina bifida, the meningomyelocele, all these questions are repeated every year. Okay, the neural tube defect. So if the whole length of the neural tube remains unclosed, then it results in your the posterior echichiasis. Now what about the anencephaly? Okay, now the neural tube, if it remains open in the anterior neuropore, okay, so non-closure of your anterior neuropore, anterior neuropore and you have a posterior neuropore. If there is non-closure of the anterior neuropore, then there will be it will result in a anencephaly, a condition known as anencephaly. Here the brain tissue also degenerates. Okay, so this was a picture given in one of the FMGA exam, and they have asked what is the condition. We will discuss the MCQ later, but this is because anencephaly is because of the non-closure of the anterior neuropore. Now let us come to the spina bifida. Now what is spina bifida? So this is due to the non-fusion of the neural tube. Okay, it is due to the non-fusion of the neural tube, and it is associated with your either the non-closure of the cranium, it is known as cranium bifidum, or non-closure of the vertebral canal, that is the spina bifidum. So you have a cranium bifidum if there is non-closure of the cranium, and you have a non-closure of the vertebral canal, that is the spina bifida. Due to the result of the non-fusion, the overlying bones, the neural tissues lie outside the cranial cavity or the vertebral column. Now when it happens in the region of the brain, it is known as your encephalocele, or it is known as the meningoencephalocele. So if it occurs in the brain, then you have to add the word encephalocele. Okay, encephalocele or meningoencephalocele. Now when it occurs in the spinal region, it is known as the myelocele or the meningomyelocele. Now how to differentiate between the myelocele, the meningomyelocele, and the spina bifida occulta. Okay, so you have types of spina bifida. So you have the spina bifida occulta, okay, the meningocele and the meningomyelocele. Okay, now in the spina bifida occulta, already there will be the problem of the vertebral bones. Okay, so there will be you will just see a minor bulge and a tuft of hair which will be grown in that area. Okay, only the bone will not close properly, the vertebral column bones will not close properly, and you will have a tuft of hair in that region, that is the spina bifida occulta. Now if there is a bulge, in this area, okay, if there is a bulge in this area and this area is filled with fluid, that is a seminal fluid, only fluid is present, then the answer is your meningocele. Okay, then the answer is a meningocele. And if there is your nerve, okay, the spinal nerves involved, okay, अगर spinal nerves involved हो रहा है, you can see the spinal nerves involved in this bulge. Okay, in this bulge, if the spinal nerves are involved, then you have the myelomeningocele. Okay, then it is known as a myelomeningocele. So you have the spina bifida occulta, where there will be a slight bulge, but there will be only the non-fusion of the bones, and you will have a tuft of hair, that is the spina bifida occulta. If they give you a question on spina bifida occulta, they will obviously point in towards the tuft of hair. Then you have the spinal fluid. They will mention about only the fluid if they have given about the meningocele. And if there is nervous tissue in that bulge, then it is known as your myelomeningocele. So these are the types of your spina bifida. Okay. Now I want a quick answer from this question. What is the embryological basis of the following anomaly? So first of all, they have given this image, okay, and they have given non-closure of entire neural tube, non-closure of anterior neurophore, posterior neurophore, and non-closure of skull bones. So anybody who wants to answer this, just now we have discussed what is this condition and what is the answer. In the chat, anyone who wants to answer this question, what is the embryological basis of the following anomaly? पहली ये anomaly क्या है? Anybody who wants to answer in the chat, what is this anomaly? Yes, you are right. It is your non-closure of your anterior neurophore. And what is this anomaly? This anomaly is your anencephaly. Anencephaly. Okay. This condition is anencephaly, and we know that anencephaly, A for anencephaly, A for anterior neuropore. Always try to remember A for anencephaly, A for anterior neuropore. Okay, so this is a correct answer. Okay. Now what is this? Which of the following condition is shown here? Anybody who wants to answer this, can you see this image properly? पहले देख लो, what is this image? What is this condition? and then you try to answer. What is this answer? Yes, the answer is your craniorachichisis. Why I am saying it is craniorachichisis? Because in this image you can see properly. Let me show you the other. Entire neural, the whole length of the neural tube, the whole length of the neural tube is open. Okay, there is no closure of anterior posterior. Entire length is open. So this is a condition of your craniorachichisis, or it is also known as the posterior rachichisis. ठीक है, craniorachichisis or posterior rachichisis. Okay, very good, everybody has answered it. ठीक है, no, it is yeah, it is the condition of your craniorachichisis. Now, which of the following condition is shown in this image? Anybody who wants to answer this? They have marked, if you can see, there is a tuft of hair. So what is this answer? There is some tuft of hair in this region. I want an answer, quick answer from this image. Anybody who wants to answer this? Just now we have studied about the different types of spina bifida. So ये क्या है? Tuft of hair. I told you, you will see a slight depression and a tuft of hair. So that will be your spina bifida occulta. Yes, you are right. Spina bifida occulta. Tuft of hair is spina bifida occulta. Very good. This spina bifida occulta. Definitely you are going to get one question from the neural tube defect in this December session. Okay, you are going to get an so you have to be very clear about the entire part. So you know that spina bifida occulta. Okay, the outer part of the vertebra is not completely closed and you will have only a slight lesion or a tuft of hairs. Oh wow, somebody has written I have passed in FMGE in the very first attempt. Very good. Okay, very good. Okay, now we have the spina bifida occulta. Okay, now also this type of questions are repeated in NEET PG as well. Okay, yes, neural tube defect is very common in INISET, NEET PG and FMG. Every time they will repeat this. Okay, now let us come to the main brain stem, the brain. We have studied about the neural tube. So they have asked the question that pons is derived from prosencephalon, metencephalon, myelencephalon and mesencephalon. Now what are this prosen, meten and myelin meson? Okay, so before that we have to study about the development of brain. So we know that till now we have studied about a neural tube, how there is anterior neuroport, a posterior neuroport, both are fusing then forming a tube. Then there will be the bulge, okay, the upper part that is forming the brain, and then there will be the lower part forming the spinal cord. Now there are certain flexures and bulges. Okay, so you can say this, the first bulge is the prosencephalon, then you have a second bulge that is the mesencephalon, and below you have the rhombencephalon. Okay, so you have PMR, prosencephalon, mesencephalon and rhombencephalon. Okay, the prosencephalon, the brain that is again divided into parts. So you have prosencephalon, the mesencephalon is the midbrain and the rhombencephalon. Now prosencephalon is again divided into telencephalon and diencephalon. Okay, then mesencephalon is directly forming the midbrain, and rhombencephalon is again divided into RMMM. Okay, metencephalon and myelencephalon. Always try to remember that wherever there is the Y in the M, it will be at the end. Okay, always I try to tell the students about this trick, that if you are confused between metencephalon and myelencephalon, so if you have a Y, myelencephalon, it will be in the end. XYZ, right? Myelencephalon, it will be in the end. So you have the prosencephalon that is giving rise to your telencephalon and diencephalon. Mesencephalon is the midbrain. Rhombencephalon is divided into metencephalon and myelencephalon. Wherever you have Y, it is the end. Okay, that is the last. Now, from the telencephalon, okay, now from the telencephalon, you have the cerebral hemisphere, okay, and the lateral ventricle. And from the diencephalon, you will have the thalamus, the third ventricle and the hypothalamus. That is from the diencephalon, with thalamus, third ventricle and the hypothalamus. Midbrain, you will have the cavity, that is the cerebral aqueduct, as you can see here, aqueduct of Sylvius. that is present in the mesencephalon. The cavity of the diencephalon is the third ventricle, and the cavity of the telencephalon is the lateral ventricles. These are the cavities. Okay, so cavity of the telencephalon is the lateral ventricle, diencephalon is the third ventricle, midbrain mesencephalon is your cerebral aqueduct. Then in the rhombencephalon, you can see you have a cavity, that is the fourth ventricle. Okay, and from the rhombencephalon you have four ventricles, cerebellum, pons, and medulla oblongata. Okay, now let us come to this chart. Prosencephalon, forebrain. Now prosencephalon, as I already told you, that you have a telencephalon which is giving rise to your cerebral cortex, corpus striatum, and the cavity is your lateral ventricle. Diencephalon, you will have thalamus, hypothalamus, metathalamus, subthalamus, cavity is your third ventricle. Mesencephalon, midbrain, the cavity is your cerebral aqueduct. And rhombencephalon, where you might get confused, I told you, wherever you have Y, that is the end. So the end portion of the brain is the medulla. So myelencephalon is for the medulla, and metencephalon, T पहले आता है, T पहले आता है, Y बाद में आता है ना? T, meten is the pons and cerebellum, and Y, the last part, is the medulla. So pons and cerebellum from the metencephalon, and medulla oblongata from the myelencephalon, and cavity is your fourth ventricle. So any confusion in this part, you may ask in the chat. So now we know that from the rhombencephalon you have two confusing things, that is the metencephalon and the myelencephalon. If you have a Y in the M, that is on the last part, and you know the entire last part of the brain is the medulla. So myelencephalon is for medulla and metencephalon is for pons and the cerebellum. Okay? So let us come to this question again. Can you answer this? Pons is derived from. Pons is derived from. Any answer in the chat? Very easy, just now we have discussed. Prosencephalon तो नहीं है, mesencephalon नहीं है. Okay, now you're confused between the metencephalon and myelencephalon. Just now I have told you, wherever you have Y, that is the last part. Okay, and T, this is the pons and cerebellum, and this is your medulla. Okay. Okay, if you have a Y in the M, that will be the last part of your nervous system, that is the medulla. So the answer, okay, the answer here will be your metencephalon. The answer here will be your metencephalon. Just now we have studied. Myelencephalon is the last part. Myelencephalon is the last part that will give rise to your medulla. Okay, wherever you have a Y, wherever you have a Y, that is the last part. अगर T है, then pons and cerebellum, Y. Whenever you have Y in the M, that is the last, X, Y, Z, that is the last part. The last part of the brain is the medulla. उसके बाद तो you will get the spinal cord. So that is the metencephalon. Pons is derived from metencephalon. Okay. So very easy to understand. पहले आता है PMR, prosencephalon, mesencephalon, and rhombencephalon. Prosencephalon मतलब your forebrain. Okay, it is again divided into your prosencephalon is again divided into your telencephalon and diencephalon. Telencephalon will give rise to the cerebral hemisphere, and the cavity is your lateral ventricle. Diencephalon will give your thalamus, hypothalamus, subthalamus. Cavity is third ventricle. Mesencephalon is the midbrain, and the cavity is your cerebral aqueduct of Sylvius. Rhombencephalon is the hindbrain. The cavity is the fourth ventricle. Rhombencephalon is divided into your metencephalon and myelencephalon. Wherever you have the word Y, okay, that is the last part. Y पहले लास्ट में आता है, T पहले आता है, ठीक है. The last part, myelencephalon, the last part is your medulla oblongata, and metencephalon is giving rise to pons and cerebellum. So this is what we studied about the development of brain. ठीक है. This one we have already attempted. Notochord is formed by, so we know that notochord is formed by primitive streak. It is from the epiblast. Now next, let us come to the cerebellum. We have so much studied about the cerebrum and the development. Let us come to the cerebellum. How many lobes are present in the cerebellum? Anybody who wants to answer this? Very easy, but a very tricky question. Just see here, they have mentioned cerebellum, not cerebellar hemisphere. So what is the answer here? How many lobes are present in cerebellum? Very easy and tricky question. Anybody who wants to answer this? How many lobes are present in cerebellum? Anyone in the chat? How many lobes are present in the cerebellum? Okay, so let us study a little bit about the cerebellum. Now you know that cerebellum has certain fissures. Okay, it has certain fissures. One is the primary fissure. Okay, one is the primary fissure which is running across the superior surface and dividing it into three lobes. Now you have got to know that you have three lobes in the cerebellum. Okay, so first is the primary fissure dividing across the superior surface and dividing the cerebellum into your three lobes. It is also known as fissura prima and it is the deepest fissure. Then you have a lateral fissure on the inferior surface and a horizontal fissure which will divide into upper and the lower halves. Okay, so we can see here, this is the primary fissure. This is the primary fissure. Okay, this is the horizontal fissure. And you have a lateral fissure, posterolateral fissure, and it is dividing the entire cerebellum into your three lobes. Okay, you have three lobes. So what are the lobes and what are the parts of the vermis and parts in the hemisphere? The lobes are your, one is the anterior lobe, one is a posterior intermediate or middle lobe, and one is a flocculonodular lobe. The flocculonodular lobe is the smallest lobe. So as you can see here, okay, here you can see that this part is the anterior lobe. Okay, this is the anterior lobe. Then this entire part is the posterior or the middle lobe, the largest part, and the smallest part is the flocculonodular lobe. Okay, and then you have certain structures corresponding to that lobes. Okay, you have certain structures corresponding to that lobes, and those are your known as vermis. This is the vermis. Okay, these are the vermis, the midline structure. So in the anterior lobe, you can see that you have the central lobule and you have the lingula and the culmen. Okay, so in the anterior lobe, in the vermis, okay, in the vermis, as you can see here, in the vermis, in the midline structure, you have the lingula. Okay, then you have the culmen and the central lobule. These are in the parts of the vermis. Okay, then in the posterior, that is the middle lobe, you have declifolium, tuber, pyramid, and uvula. Okay, and in the flocculonodular lobe, you have the nodule. So these are the parts of your vermis. Now the question was, how many lobes do you have? How many lobes do you have in the cerebellum? Now I want a correct answer from this. How many lobes do you have in the cerebellum? They have given 2, 4, 6, 8. What is the correct answer? How many lobes do you have in the cerebellum? Just now we have studied. Any answer in the chat? You know that you have 3 lobes in the cerebellar hemisphere. And in cerebellum, you have two cerebellar hemisphere. So they have asked the question, cerebellum. So 2 cerebellar hemisphere means you have 6 lobes total, both sides. What, 3 on this side, 3 on that side. So the answer will be your 6 lobes. Okay, it's a tricky question. They have asked you about cerebellum. Okay, if they would have mentioned about cerebellar hemisphere, then the answer is your 3 lobes. If they have mentioned about cerebellum, then you have 2 cerebellar hemisphere, one on this side, one on that side. So you have 6 lobes. Okay, so the answer is 6 lobes. Many of the students might say that the correct answer is not in the option, but you read the question properly. They have mentioned about cerebellum as a whole. Okay, in one cerebellar hemisphere, if you can see here, you have three lobes. Okay, so you can see the anterior lobe of this part and the posterior lobe of this part and uh, the flocculonodular lobe. Okay, this side, another side you have same thing. So you have total 6 lobes in the entire cerebellum. Okay? If they would have given the option as cerebellar hemisphere, then the answer would be 3 lobes. Okay, so it is a tricky one. So that's about the cerebellum. So again, you can see here in this region that you have a fissure, primary fissure. This is a primary fissure, which is very deep. It is known as a fissura prima. It is known as the fissura primera. Then you have the horizontal fissure and you have a posterolateral fissure in this area. Okay, so that is about the lobes and the fissures. Okay, now what are the nucleus in the cerebellum? So you have certain nucleus. Let us study a little bit about that. You have the vestigial nucleus, the emboliform and the globus nucleus, and the dentate nucleus. The vestigial nucleus are mostly, you can see that they are the vestibular apparatus through the vestibular nerve, emboliform for the paravermal region or the spinocerebellum, and dentate from the neocerebellum. Now what is important is the location. It is important that you should know where the nucleus are present. So as you can see here, the vestigial nucleus are present the most medially. Okay, the most medially you have the vestigial nucleus. Then you are going to have the globus nucleus. Okay, uske baad you will have the emboliform nucleus, and this structure, if they have given this structure, this is the dentate nucleus because it has many dentation. Okay, so these are the location. Okay, sometimes they might ask in a cadaveric image as well. Okay, so they might put a point on the most medial structure. So the most medial structure here is the vestigial nucleus. Okay, after vestigial nucleus you have the emboliform nucleus. Okay, and sorry, you have the globus nucleus. First you have the vestigial nucleus here. Okay, then you will have the globus nucleus here. Then you are going to have the emboliform nucleus, and the one day if you give this kind of structure, if they give this kind of structure, which you can see here, this gray matter. Okay, this kind of structure, that is your dentate nucleus. Okay, so if you see this arrangement, you can see that the most medial nucleus is the vestigial nucleus. Okay, okay, now we have certain intrinsic neurons in the layer of the cerebellar cortex. So you know that what are the layers, as you can see in the cerebellar cortex. Similarly, in the cerebellum you have the outer gray matter and the inner white matter. So the most outer layer is the molecular layer. Okay, the most outer layer is the molecular layer. Then you have the Purkinje layer, then you have the granular layer, and then you have the white matter. In the white matter you have the cerebellar connections. Those are the efferent fibers, those are the climbing and the mossy fibers, and you have the efferent fibers, that is axons of the Purkinje. These are your white matter. Okay, and in this layers you have certain cells. So in the outer molecular layer you can see a cell, that is your stellate cell. Okay, in the Purkinje layer obviously you will get the Purkinje cell, and in the granular layer you are also going to get the granular cells, the Golgi cells. Okay, the Golgi cells, the granular cells. Okay, so these are the cells. The molecular layer you are going to have the stellate and the basket cells. In the Purkinje cells, and in the granular you will get the granular cells and the Golgi cells. Okay, so these are the layers and the cells. So a question came on this that cerebellar cortex, which of the following cells? So they have given pyramidal, stromal, oligodendrocytes, and the Purkinje cells. So I want a quick answer from you. Cerebellar cortex has which cell among this? A quick answer. So just now we have studied that you have a Purkinje layer. So you have the Purkinje cells. Okay, in the cerebellar cortex you have the Purkinje cells. Okay. Which is the smallest lobe of the cerebellum? The flocculonodular, the lingular lobe, the pyramidal lobe, and the archicerebellum. Any answer in the chat? This is very, very easy. I have just now repeated. Which is the smallest lobe of cerebellum? Very small at the lower end you have like this. What is that lobe called? Smallest lobe of cerebellum. Anybody? So the answer is your, yes, the answer is A, that is your flocculonodular lobe. Okay? The smallest lobe of cerebellum is the flocculonodular lobe. Many questions came on cerebellum, so there is high chance that you might get a question on cerebellum as well in the December FMG. Okay, so cerebellar cortex, Purkinje cell, smallest lobe, flocculonodular. Now next question is most medially located nuclei of cerebellum. So they have given lentiform, fastigiae, putamen and caudate. So the options are so obvious that you know the answer. Lentiform, putamen and caudate. ये तो है ही नहीं cerebellar nucleus का part है ना. So the answer is a fastigiae. Just now I have told the fastigious nucleus is most medially located. As you can see here, the most medial is the fastigiae, then you have the globus, then you have the emboliform, and then you have the dentate nucleus. Sometimes they might give a arrow on the dentate. Okay, in the cadaveric image they might give a arrow on the dentate and ask you which is the correct nucleus. So the one where you see a dentation, a curve like dentation, that is dentate, and the most medial is your fastigial nuclei. The most medial is your fastigial nuclei. Yes. Okay, now let us come to the tracks. Okay, let us come to the tracks. Which of the following tracks is seen in the posterior column of the spinal cord? Posterior column of the spinal cord. So they have given your lateral spinothalamic, fasciculus gracilis, cerebrospinal, posterior spinocerebellar. Any answer from the chat who has studied about the tracks of the spinal cord? Which of the following tracks is seen in the posterior column of the spinal cord? Any answer? Who wants to answer this? Very easy. If you understand it, then it is very easy for you. Any answer from the chat? Lateral spinothalamic? No, it's not lateral spinothalamic. Pehle, let us come to the tracks. Okay, now you know that this is the spinal cord. Now what you can see in the spinal cord? You can see a butterfly. Let us study it, then you will answer it. Okay. You can see a butterfly shaped gray matter. It's a butterfly shaped gray matter. Okay, this is the butterfly shaped gray matter. You know that spinal cord ulta hai brain se, so the white matter will be outside, the gray matter will be inside. Then the outer areas are white matter. Now this area is known as the posterior funiculus. They are known as posterior funiculus. This area is known as the lateral funiculus, and this area is known as the anterior funiculus. Okay, now you know that this anterior white commissure is the area where there is crossing over of the fibers. Okay, so that is about a funiculus or the column. Okay, now we'll come to the ascending and the descending tracks. Okay, now you know that there are certain ascending tracks which are sensory. So you have certain tracks here. Okay, these are also known as lemniscus system. Okay, so if you see in the dorsal part, dorsal means the posterior part, dorsal column, medial lemniscus system, you have fasciculus gracilis and fasciculus cuneatus. Okay, you have fasciculus gracilis and fasciculus cuneatus. Okay, then spinocerebellar tract is in the lateral column. You have the posterior spinocerebellar and you have the anterior spinocerebellar. Okay, and anterolateral system, you have the spinothalamic tract. Okay, you have the anterior spinothalamic, lateral spinothalamic and the spino-olivary fibers. These are about the ascending fibers, those are sensory. And if you see about the descending fibers which are red in color, so you have the lateral corticospinal cortex, it will descend. So wherever there is corticospinal, it is descending. So you have the pyramidal tracts. Those are the lateral corticospinal and the anterior corticospinal. And then you have the extrapyramidal tracts. Those are rubrospinal, reticulo, olivulo, and vestibulospinal. Spinal word last में है, that is those are the descending tracts. So this is the table. Now you can see here that ascending and descending tracts. So you have the posterior column or the posterior funiculus. So in the posterior or the dorsal part, you have fasciculus gracilis and fasciculus cuneatus as ascending, and descending are your septomarginal, and you have the fasciculus interfascicularis. Okay, it is also known as the semilunar tract or comma tract of your Schulze. Okay, those are the descending tracts in the posterior column. What about the lateral column? Now lateral column, you have all the spinocerebellar tract. You have the posterior spinocerebellar, anterior spinocerebellar, and the lateral spinothalamic. Also you have spinotactal, olivary, and ischius tract. Okay, and descending are all the corticospinal, rubrospinal, okay, cortico-rubro, and reticulospinal. Okay, those are your descending tracts. And in the anterior part, you have the spinothalamic tract, anterior spinothalamic tract, and the spinoreticular tract. And if you see the descending tract, you have the again the corticospinal, vestibulospinal, tectospinal. These are the descending. So if they are asking about the dorsal column, that is a posterior column, then you have two tracts, that is fasciculus gracilis and fasciculus cuneatus. Okay, so let us come to the question again. So they have given the lateral spinothalamic, it is wrong. The rubrospinal, that is also wrong. The posterior spinocerebellar, it is also wrong. So I told you that in the posterior column of the spinal tract, you have the fasciculus gracilis and the fasciculus cuneatus. Okay, so on the posterior part, you have fasciculus gracilis and fasciculus cuneatus. Okay. So spinothalamic, if they have told about lateral spinothalamic, it is in the lateral column. Okay, the lateral spinothalamic is in the lateral column, and the anterior spinothalamic is in the anterior column. Okay, very easy. Anterior spinothalamic is in the anterior column, lateral spinothalamic is in the lateral column, and spinocerebellar, the posterior and the anterior spinocerebellar, those are also in the lateral column. So posterior column, only two tracts you have to remember, important for the exam, that is your fasciculus gracilis and fasciculus cuneatus. I hope it is clear now. Okay. So those are the tracks mostly I have shown in this image. You can see these are the descending tracks. Okay, so the cerebral cortex to your spinal cord, these tracks are your descending tracks. Okay, so you can see that this is the corticospinal, then you have from the tectospinal, the rubrospinal, okay, then you have the vestibulospinal, and then you have the reticulospinal. These are your descending tract. And ascending tract, as discussed, you have the spinothalamic. As you can see here, the lateral spinothalamus, it is going to the thalamic, then the anterior spinothalamic tract. Okay, then you have the fasciculus gracilis, the fasciculus cuneatus, okay, the spinotactal, the spino-olivary. These are all your ascending tracts. Okay, now you know that the ascending tracts are your sensory and the descending tracts are your motor. Now there is a question on this, that the left side spinal lemniscus contains which fibers? The left side spinal lemniscus contains which fibers? They have given left side pain touch temperature, right side pain touch temperature, right side position, vibration and sense, and left side position, vibration and sense. So any answer from the chat, if you want to answer this, what is the option, correct answer to this? What is spinal lemniscus, and the left side will contain what type of fiber? Any answer? Want to attempt this question? What is spinal lemniscus, and what kind of fiber do you have? So you have answered right side, pain, touch and temperature. Yes, you are right. The answer is your right side, pain, touch and temperature. Now let us come to the, no, it is not left side, it is right side. It is right side, pain, touch and temperature. So let us come to this, what is lemniscus and all. So you can see here, there are certain order of neurons, you have different order of neuron. You have the first order neuron, okay, then you have the second order neuron, and then you have the third order neuron. Now the first order neuron are on the same side. The second order neuron, as you can see, the second order neuron are crossing. As I told you, there will be the anterior commissure where they will be crossing over of fibers. So the second order neuron will be crossing. So anything which is related to the second order neuron will be of the opposite side. Now you know that anterior spinothalamic tract is mostly for the crude touch and pressure, and the lateral spinothalamic tract is for temperature and pain. Okay, the temperature and pain, these are mostly the sensory fibers we are talking about. So the lateral spinothalamic tract, it is mostly because of the pain and the temperature, okay, pain, temperature and light touch, okay. And the dorsal column is mostly responsible for your deep touch, proprioception and vibration. So if they would have asked about the dorsal fibers or the posterior fibers, it is for the deep touch, propulsion and vibration. For the lateral part, it is your pain, touch, pain, temperature and touch. And these are mostly the second order neurons which are going to cross in the anterior commissure, okay, they are going to cross. So the question here was the left-sided spinal lemniscus. So this is, what do you mean by the spinal lemniscus? So this is what do you mean by the spinal lemniscus, okay. The left side spinal lemniscus, they are mostly going to cross here. They are the second order neuron, okay. They are the second order neuron, and it is responsible for your pain, touch and temperature. So since it is left side, the answer will be your right side, pain, touch and temperature, okay. So clear? Any doubt here? Spinal lemniscus of the left side will cross towards the right, okay, the fibers will be crossed in the anterior commissure, and the spinal lemniscus are responsible for pain, touch and temperature, okay. This spinal lemniscus is responsible for pain, touch and temperature, and since they will be these are the second order neurons, they will be crossing over, and crossing over will lead to your opposite side. So right side, pain, touch and temperature. It contains right side, pain, touch and temperature. The answer will be this one. Okay, so I want to do a quick recap before I end this session today. Let us do a quick recap of what we have studied. Okay, so we have started with some embryogenesis. So now you know that how the blastocyst is formed. So first we have seen that there are different cell stage, 2 cell stage, 4 cell stage, 8 cell stage and morula. Then you have differentiations of the cell into the outer cell mass and the inner cell mass. So now you know that, pehle you know that there is a zona pellucida, okay, you know that there is a zona pellucida. Now this will disintegrate, the zona pellucida will disintegrate, and now you already have this naked mass, okay. And once the zona pellucida disappear, you have this naked mass, the outer cell mass, okay, and the inner cell mass. So this will again undergo compaction, and then you will have a cavity form, that is the blastocele. This is the blastocele, okay. And then you have one side aggregation of cells that is known as the inner cell mass, and the outer cell mass, that is the trophoblast. Now you know that the trophoblast is ultimately going to give rise to your the cytotropoblast and syncytotropoblast, and then they are taking part in the formation of your placenta, right? Now what is important is that the inner cell mass. Okay, the inner cell mass. So you know that the inner cell mass at second week it will undergo the bilaminar germ disc. Okay, bilaminar disc in the second week. So next, sometimes they might ask you about the bilaminar disc. Okay, a question might come on the bilaminar disc. So that is on the second week. The rule of 2. Okay, if you have a bilaminar disc, that is rule of 2, and it is in the second week. Trilaminar disc means that is the third week. So you have a upper columnar layer that is known as the epiblast, and a lower cubical layer, that is known as the hypoblast. And then you have the first cavity C1, this is your the yolk sac. Okay, now all the layers are developing from the epiblast, that is the ectoderm, endoderm and mesoderm. Okay, so you can see after this you will have another cavity form, that is the C2, cavity number 2, and that is known as the amniotic cavity. Okay, you have an amniotic cavity, and C1 as shown earlier will form the yolk sac. Okay, and the hypoblast cells are going to migrate. They are going to migrate towards the yolk sac. Okay, so we know that the extra embryonic endoderm is formed from the hypoblast. Okay, that is giving rise to your yolk sac. And then you have a cavity, amniotic cavity. After that, what happens? There will be the formation of your trilaminar disc. Okay, that is your gastrulation, and this is in the third week. Okay, trilaminar disc, gastrulation, third week. And then what is important in this area, that in the gastrulation you have a rod like structure form, and that is your primitive streak. Okay, primitive streak, what we are concerned with is the primitive streak. Okay, primitive streak. So after this primitive streak is formed, what we are going to do is that we will take a cut section here, and we are going to remove the amniotic cavity, and we are going to look the layers from above. Okay, we are going to look the layers from above, and that will result in your structure like this, that is known as your embryonic disc. Okay, so we have already studied now the embryonic disc. In the embryonic disc we can see the columnar, epiblast and the cubical hypoblast. So there is a formation of a procaudal plate. Okay, there is a formation of a procaudal plate. Okay, in the head, in the cranial end, and in the tail you will have the formation of a primitive streak. In the tail end you will have a formation of a primitive streak, and in the head end you will have a formation of your procaudal plate. Okay, now we want to know that how the notochord is formed. So it's very easy if you understand. So you know that there is a procaudal or precaudal plate which is going to form the future mouth, and then you will have a cloacal membrane or the anus. Now you have the primitive streak, okay, which is also going to give rise to a node that is known as the primitive node. So sometimes they might ask a question on your Hansen's node. Okay, Hansen's node. Okay, so you have a primitive node and a primitive streak and a primitive groove, and that groove is also going to give rise to a primitive bead. Now from this node, certain cells will migrate towards the procaudal plate to form a solid structure first. This solid structure is known as your notochordal process. notochordal process. Now this solid structure will undergo canalization, that is known as notochordal canal. Then you will have the neuroenteric canal. Then you are going to have a plate that is known as the notochordal plate. Notochordal plate. And after that only you will have the formation of your definitive notochord. Okay, definitive notochord. Okay, so till this much it is clear. Now I want to ask the question, if you can answer this. Just now we have studied that Hansen's node. This came in NEET PG, okay, so it might also get repeated in the FMGE as well. Hansen's node is derived from Hansen's node is derived from So I want a quick reply from this the chat. If you know this, Hansen's node is derived from. Anyone who wants to answer this? Hansen's node is derived from. Now there are two confusion things in this question. Hansen's node is derived from. Now you know that endoderm तो नहीं है, hypoblast भी नहीं है, but you're confused between mesoderm and epiblast. Okay, so you know that the primitive streak, we are confused between the mesoderm and epiblast. No, Hansen's node, no, the answer is not B. Hypoblast is not giving rise to a Hansen's node. Hansen's node or primitive node, just now I have told you, primitive node is also known as Hansen's node. Okay, primitive node is also known as Hansen's node, and primitive node is developing from your primitive streak. Primitive streak, है ना? Primitive streak. Now primitive streak is arising from your epiblast. So the answer will be your epiblast. Okay. Don't confuse Hansen's node with your Hübner's membrane. Okay, there is a membrane which is known as the Hübner's membrane. So you are getting confused with that. That is not. So there are two H in this embryology. One is a Hansen's node, one is a Hübner's membrane. So the answer is your epiblast. Okay, the answer is your epiblast. Very easy. And another question I will ask, that is your. Let us discuss that question. Just now I have told you remnant of notochord. So you have the remnant of notochord. Let us write very easy. I have given a very easy option to you, that these two ligaments तो है ही नहीं. Okay, so the answer is your nucleus pulposus. Okay, the answer is your nucleus pulposus. Now one more tricky thing they might ask, that they might not give you the option of nucleus pulposus, as it is very obvious. They might give you the option of your apical ligament of Dens. Okay, among all the other ligaments, they might give you the option of your apical ligament of Dens. Then the answer will be the apical ligament of Dens. So you know that the remnant of notochord is both the nucleus pulposus and the apical ligament of Dens. So sorry, this is a very favorite question. So nucleus pulposus and apical ligament of Dens. Now third question. neural crest cells are derived from. So I am giving the option: epiblast, hypoblast, mesoderm, ectoderm. Now I want a correct answer from this question. Neural crest cells are derived from epiblast, hypoblast, mesoderm, ectoderm. What is the, you might get confused with two options. Yes, you are right, epiblast. Now see why I want to discuss this question, because hypoblast, mesoderm ये तो नहीं है. You might get confused with epiblast and ectoderm. Okay, now you know that neural crest cells are derived from the neuroectoderm, है ना, neuroectoderm, but this ectoderm is ultimately getting originated from epiblast. So you know that epiblast is giving rise to all the three layers, that is ecto, meso and endo. So the correct answer here will be the epiblast. Okay, if they would have not given the option of epiblast, then you could have marked the answer neuroectoderm, but here they have mentioned about yes, ectoderm is derived from epiblast. So the correct answer, if they have given ectoderm and epiblast, directly don't mark ectoderm, you have to mark epiblast. Every time they are asking this and you're getting confused. Now one more question I want to ask. Wait, first let us see the this chart, okay, which I have made by seeing all the books. Okay, what I understood from Langman. I'm mostly following the Langman book of anatomy for embryology. Okay, so Langman has all the correct things written there. So you have the blastocyst, you have the inner cell mass and the trophoblast. Now inner cell mass, again I have divided into the epiblast, where you have the embryonic epiblast and the amniotic ectoderm. Okay, and the hypoblast, it is giving rise to your extra embryonic endoderm. Okay, now a question might come on this, that extra embryonic endoderm is derived from. So they will give the options as epiblast, hypoblast. Okay, then they might give you trophoblast as well, or they might give you the option as your some other options. They might give you epiblast, hypoblast, trophoblast, or they might also give you some other options like ectoderm, just extra option. So now you know that these are completely wrong. You are confused. You know that endoderm, whenever you see the word endoderm, you try to remember that epiblast gives all the three layers, that is ectoderm, endoderm and mesoderm. But the trick here is they have asked about the extra embryonic endoderm. Okay, so don't directly mark the answer as epiblast here. You have to read the question properly. Okay, you have to read the question properly. So the answer here will be your hypoblast. Yes, the answer will be hypoblast, because they are asking about the extra embryonic endoderm. Okay, this is also a question asked from the NEET PG. Okay, they have asked NEET PG. So extra embryonic endoderm is derived from hypoblast. Okay, extra embryonic endoderm is derived from the hypoblast. And if they have asked, the next question will be on the embryonic endoderm. So if they will ask about embryonic endoderm, this is from your epiblast. So don't come with a doubt. Many students used to come to me with a doubt that, ma'am, you told that epiblast give rise to your ectoderm, endoderm and mesoderm. So we marked the answer as epiblast. But you have to see the question. They have mentioned about extra embryonic endoderm. If the word is extra embryonic endoderm, then it is derived from hypoblast. And if they have mentioned the word embryonic endoderm, then it is derived from epiblast. Okay, you have to see the question properly. Okay, So then you have the primitive streak, and then you have the mesoderm as well. So extraembryonic mesoderm, embryonic mesoderm, and the notochordal process. Now in some books, in some questions, they have mentioned about this question as well. Let us discuss this doubt as well, that notochord is derived from. Okay, so again they have given the option as epiblast, hypoblast, mesoderm, and endoderm. Now they have mentioned mesoderm and endoderm. So what is the correct answer here? Okay, here also you might get confused, because you know that it is not derived from hypoblast, it is not derived from endoderm, but you might get confused with the epiblast and the mesoderm. Okay, now you know that notochord is derived from the primitive streak. Okay, it is derived from the primitive streak. Primitive streak is derived from the epiblast. You have to mark the answer as epiblast, but this is, there is a different, they have write derived from, so the answer will be epiblast. Okay, now if they ask you notochord is of which origin, so here if they give you ectoderm, endoderm, and mesoderm, or if it is derived from which germ layer. Okay, now they have changed the question and they have asked you about which germ layer, and they have given you ectoderm, endoderm, and mesoderm. Now they have correctly mentioned a germ layer, so then the answer is your mesoderm. Okay, then the answer is a mesoderm. So if they have mentioned about the germ layer, then it will be mesoderm, and if they have mentioned epiblast, hypoblast, and then mesoderm, and notochord is derived from, then it will be your epiblast. Okay, so there is a change of pattern in the question. If they mention about the germ layers, okay, then it is your mesodermal in derivative, mesodermal in origin. Okay, I hope this is clear now. Okay, so you try to remember this chart which I have drawn, because it is seen from Langman book. Okay, the book Langman has very properly described about it in paragraphs, so I have just made a chart out of that. So you try to remember this chart. I think it will help you in your exam, because mostly you might get a question on your neural tube, neural crest cells. Okay, neural crest cells and notochordal process, very confusing, and they are every time asking origin of epiblast and hypoblast. So this chart is very important for your exam. Okay, I want you to just write in a page this chart, and then remember, then you can recap how to remember the origin and the derivative. Okay, so that is all. Then we have also studied about the neural tube. You know that there is anterior neuropore, posterior neuropore, and they might ask you a question on this also. The date 25th day, and this is the 27th day. And rachichisis, you know that the whole length of the neural tube is unclosed. You will see the whole length. Okay, rachichisis also repeated, craniorachichisis or posterior rachichisis. That anencephaly, as asked every time in FMGE, so you know that non-closure of the anterior neurophores. Always try to remember AA. A for anencephaly and A for anterior neuropore. Okay, then you know that spina bifida. Okay, so if there is occulta, then there will be tuft of hair. If there is only fluid, then it is spina, then it will be meningocele, and agar nerves hai, so it will be mylomeningocele. Okay, so spina bifida is also asked every time. Okay, so these are the questions. Then we have studied about the brain, which I am again going to recap once. You know that there are three swellings and certain flexures. So you have the prosencephalon, you have the mesencephalon, and the rhombencephalon. PMR, okay, PMR, physical medicine and rehabilitation, one department you have, right? So that is, just try to remember PMR, that is prosencephalon, mesencephalon, and rhombencephalon. Then on prosencephalon you have telencephalon, and the cavity, this will give rise to cerebral hemisphere. Then you have diencephalon, which will give rise to thalamus. The telencephalon cavity is lateral ventricle, diencephalon you have the third ventricle. And then in the midbrain you have the cerebral aqueduct, and in the hindbrain you will have the fourth ventricle. Now what you get confused is the rhombencephalon. It is divided into metencephalon and myelencephalon. So just now I have told you that whenever you have the word Y, okay, Y is at the last, so that will include the medulla oblongata, and metencephalon will have your pons and cerebellum. So pons is derived from your metencephalon. Okay. So that's what we have studied. Then we have studied about the lobes. You know, there are three fissures: primary, posterolateral, and horizontal. You have three lobes. So if they ask you about cerebellar hemisphere, then you will write three lobes. If they ask you about cerebellum, then you will write six lobes. The smallest lobe is the flocculonodular lobe. This is the smallest lobe. Okay, this is the flocculonodular lobe. That is the smallest lobe. This lobe is the smallest lobe. Okay. Then they might ask you about a nucleus. So you know that you have the most medial nucleus, that is your fastigial nucleus. Then you have the globus. उसके बाद emboliform. Sometimes they might give a arrow on the dentate nucleus. Okay, so the dentate nucleus is this one. It is like this. Okay, that is the dentate nucleus. So that's about the cerebellum. Then we have studied about the cerebellar cortex. So you know that outer layer is the molecular, then you have the Purkinje, and then you have the granular, and then you have the white matter where you have the fibers. So you have the climbing fibers and the mossy fibers. These are the cerebellar connections and the axons of the Purkinje cells. Now what are the cells? You will have the stellate cells and the basket cells in the molecular layer. Purkinje layer will have the Purkinje cells, and in the granular layer you will have the granule cells and the Golgi cells. So these are the topics discussed. Then we have come across the white matter of the spinal cord. Okay, so you know that there is a butterfly shaped structure. Now what do you get in the gray matter? If you see the histology part, so in the gray matter you will have neurons, neuroglia, and blood vessels. Now what are these neurons? These are your multipolar neurons. Okay, these are the multipolar neurons which are more in the anterior horn than the posterior horn. Now neuroglia, you have the oligodendrocytes, you have the Schwann cells. Okay, these are the neuroglia, and you will get the blood vessels. Okay, so those are about the and you also have certain nucleuses. Okay, that one we'll discuss someday, the Clark's column and all. But now for now you try to remember that this white matter is the posterior column, this is the lateral column, and this is the anterior column. Okay, that is the white matter, and you know that the ascending fibers are your sensory. Okay, the ascending fibers are your sensory. They are the efferent pathway, and the descending fibers are your motor. Now on the posterior most part, you have the gracile tubercle and the cunate tubercle. So you have the fasciculus gracilis and the fasciculus cunatus. So most medially you will have the fasciculus gracilis, and just lateral to that you will have the fasciculus cunatus. So they have asked about the dorsal column. So dorsal column will have your fasciculus gracilis and fasciculus cunatus. On the lateral column you have the posterior spinocerebellar and the anterior spinocerebellar. So sometimes they might ask you on the spinocerebellar tract, and in the anterolateral system you will have the lateral spinothalamic and the anterior spinothalamic. Also you have the spino-olivary fibers. So these are about the columns. Okay, and if you see the descending, you have the pyramidal tract. So all the corticospinal, like lateral corticospinal and anterior corticospinal, are coming under the pyramidal, and in the extrapyramidal you have the rubrospinal, the reticulo, the olivospinal, and the vestibulospinal. These are the motor fibers. Okay, and the ascending tracts are the sensory fibers. So try to remember this diagram, then you will understand better. But the question came on the dorsal column, so it will be a fasciculus gracilis and fasciculus cuneatus. Okay, then we have studied about the lemnisci, the spinal lemnisci. So you can see there are different orders of neurons. So you have the first order neuron. Okay, the first order neuron, there is no crossing over in the first order neuron. When you come to the second order, so you can see that there's a crossing in the anterior commissure. The crossing over occurs in the anterior commissure, and then you have the third order neuron. They have asked about the lateral fibers or laterals of the spinal lemniscus. They are going to cross over in the anterior commissure, and they are responsible for your pain, touch, and temperature. Okay, and if they would have asked about a dorsal column, so it is your deep touch, proprioception, and vibration. Okay, that is the deep touch, proprioception, and vibration. Okay, if there is about the descending tracts, most are mainly because of the motor. These are the voluntary controls. Okay, and voluntary motor control in the descending tract. And if they are asking about the ascending tracts, mostly it is concerned with the pain, touch, and temperature, and that is on the lateral part. Okay, ventral spinothalamic, it is mostly for the light touch. Lateral spinothalamic because of pain touch, and dorsal column mostly the deep touch. So the question might come on a deep touch and light touch. Okay, if they have asked about light touch, then it is your ventral spinothalamic. Okay, it is ventral spinothalamic. Light touch is for the ventral spinothalamic, and if they have asked about deep touch, proprioception, and vibration, it is your dorsal column, that is your posterior funiculus, that is fasciculus cuneatus and fasciculus gracilis. So this might be another question, that is deep touch, proprioception, and vibration, and light touch. So light touch is ventral spinothalamic, and deep touch is your dorsal column, that is your fasciculus gracilis and fasciculus cuneatus. Okay. Okay, so with that I end today's topic. So most of the things are covered here, and you also need to study a little bit about the limbic system. So they have asked about cingulate gyrus, so it is a part of the limbic system. That question came in one of the FMG exams. Okay, about the cingulate gyrus, that is a part of the limbic system. And we have almost covered everything. You can also study a little bit about the thalamus and the nucleus there. Sometimes they might ask a question on thalamus as well. Okay, and you might ask on the skull bones. So I already told you about the terion, the asterion. Okay, so the terion सबसे ज़्यादा terion ही पूछते हैं. ठीक है, so try to study about the fontanelles, anterior fontanelle, posterior fontanelle, terion. ठीक है, those are the repeated questions. Okay, and when I will take the next session, I will teach you about little bit about the cranial nerve nuclei. Okay, so there are many questions on the cranial nerve nuclei, so that will be a long session, because many things has to be taught under the cranial nerve nuclei, the course of the cranial nerves, and so many questions is to come on the cranial nerve nuclei. So in next session we'll start, if I take a little bit about the neuroanatomy, we'll do the cranial nerve nuclei and the head and neck. We'll start with the head and neck as well. So cranial nerve nuclei का बहुत कुछ आता है, the sense which is sensory, which is motor, which is mixed, okay, which has the longest course, okay, which are the cranial nerve nuclei present in the pons, nerve nuclei affected in the midbrain. So many questions is to come on the cranial nerve nuclei. Okay, and in the spinal cord, mostly you are also going to be asked about the level of the spinal cord. So in the adults, where is the level of the spinal cord? In the children, what is the level of the spinal cord? So anybody who knows what is the level of spinal cord in adult, the lower level of spinal cord in adult? If you know, you can answer it. What is the lower level of spinal cord in adult, and what is the lower level of spinal cord in children? So you know the vertebral level, Anyone who knows the vertebral level, the level of spinal cord in adult and the level of spinal cord in children's. So it is mostly your the L1 in the adults and your L3 in the children's. Okay? And then they might also ask you about the level of the subarachnoid space. Okay, कहाँ है subarachnoid space का level? No, it is not L4. Okay, L4, L3, L4 or L4, L5 is the space where we give the lumbar puncture, but the lower level is at the L1. Okay, it is at the level of L1. So I think I have uh, taught you about the levels in one of the class, maybe in the neuroanatomy it has been discussed. I think I have discussed the levels of spinal cord in one of the classes, so you should go through that, okay, because they used to ask many questions on these levels. So you can see here, this is the lower level of the spinal cord, that is the conus medullaris. Okay, so it is in the level of the L1. Okay, L1. Okay, and so you can see that this is the extent of the spinal cord. Very important question. That is at the lower border of the first lumbar vertebra in adults. This is in adults. Okay, and in children it is at the level of the third lumbar vertebra. So you have to know also about the length of the spinal cord. It is 45. The length of the vertebral column is 65. Lower end is known as conus medullaris and it has 31 pairs of spinal cord. So for adults it is your L1. Okay, for adults it is for L1 and it is L3 for children's. Okay, for L3 for children's. And if they ask about the subarachnoid space or the dura mater, it is at the level of your S2, lower border of S2. So lower level of spinal cord is your L1 in adults. Okay, in adults it is L1 and L3 in children, and subarachnoid space or dura mater it is at the level of the lower border of the S2. So these two you have to remember. Okay, so this is about the spinal cord, the most asked question. What is the lower level of spinal cord? What is the lower level of spinal cord in newborn? And one more thing you might ask in the exam is the modification of the pia mater. So in the spinal cord, you know that there are certain modification, that is the ligamentum denticulata. Okay, you have the ligamenta denticulata. So it looks like teeth. It looks like teeth. Okay, then you have the ligamentum denticulata. You have the phylum terminally. Okay, you have the phylum terminally. Okay, it is also the modification of pia mater. You have the phylum terminally. Now you have till it is enclosed with the dural space, that is the phylum terminally internum, and after S2, okay, from S2 to the coccyx it is known as the phylum terminally externum. Okay, so this is also asked in the exam. Phylum terminally externum from your S2 to coccyx. Okay, and phylum terminally internum is from your L1 to S2. Okay, from L1 to S2 that is your phylum terminally internum, and from S2 to coccyx bone that is your phylum terminally externum, and these are the modification of your pia mater. Okay, so sometimes you might ask on this also. So you can see in this image, you might give a cadaveric image and ask what is this, what is the level. So this is your phylum terminally externum. Here you can see that This is the phylum terminally, okay. And if it is going down till the coccyx, it will be externum, and if it is included under the sacrum S2, then it will be internum. So this is the phylum terminally, and here you can see this is the coccyx, which is at the lower border of your L1, and these are the cauda equina, which looks like the tail of a horse. Okay, those are about the spinal cord. And one more thing, about a lumbar puncture, I want to discuss the layers. Okay, so what are the layers? Sometimes they might ask you a question on the layer which is not pierced during the epidural anesthesia or during the lumbar anesthesia. So you know that the first layer will be the skin. Then you have the superficial fascia. Then you have a ligament. What ligament is this? This is known as the supraspinous ligament. Okay, after piercing the supraspinous ligament, you have another ligament. This is the interspinous ligament. Okay, after interspinous ligament, you are going to have ligamentum flavum. ठीक है, ligamentum flavum के बाद, okay, now it is important, this space, this yellow space, let me draw with a different color, this purple space, this space is known as the epidural space, okay, and this red color which I am drawing, this red color, this is the dura mater. This is the dura mater. Okay, one more time, let me, this is the epidural, this is the epidural space, this is the epidural space, okay, and then you have another layer, that is the dura mater, okay, the dura mater, and then you have another layer, that is the arachnoid mater, okay, and then this blue color, that is your subarachnoid space, and the space between these two, this is the subdural space, if you have to remember according to that, but this is the subarachnoid space, is a space where you will get the CSF, है ना? The subarachnoid space. Okay, so you can see the needle, it is piercing through all the layers, it is piercing the subarachnoid space, it is piercing the arachnoid matter and entering into the subarachnoid space. But if the needle has to enter the epidural space, that is the purple color, then it will enter in this space and it is not going to pierce the dura mater. Okay, it is not going to pierce the dura mater. So epidural is above the dura, as I told you, so it is not going to pierce the dura mater, and if it is piercing the dura mater and the arachnoid mater and entering into the subarachnoid space, that is a lumbar puncture. So this needle is for the lumbar puncture, and if I am giving a separate needle which is entering through this epidural space, okay, this is the epidural anesthesia. So there is confusion between epidural anesthesia and the lumbar puncture. So lumbar puncture, the needle will enter into the subarachnoid space, so it will pierce the dura and the arachnoid, but in the epidural anesthesia, it is not going to pierce the dura. Epidural means epidural, which is above the dura mater. Okay, so all these ligaments will be pierced, but the dura mater will be not pierced in the epidural anesthesia. So this is epidural and this is lumbar puncture. Okay, so that's about yeah, epidural POPs. So that's about the ligaments and the structures pierced during the LP, lumbar puncture and the epidural anesthesia. Okay, so asked in the exam. So that's what I want to tell. Okay, with this I end today's topic, and I hope you understood the topics well. If you have any doubt regarding the embryogenesis, I will take a session on the embryology later on, and on the entire embryogenesis, the gastrulation, the neural deformation, the neurulation, okay, the germ layer and all. So if you have any doubt, you can ask me in the chat. I will be replying in the chat, and with this I finish the neuroanatomy part 3, and the next session will start with the head and neck and the cranial nerve nuclei simultaneously, because cranial nerve nuclei is a long part, cranial nerve course and cranial nerve nuclei, Bell's palsy, many questions to come on that. So with that I end today's topic. Any doubt you can ask in the chat, and I hope you understood the topic well. All right, So thank you very much for your patience, and any doubt, any feedback, you can just write in the chat. It will be very helpful. If I have to improve in something, you can also write that. If you didn't understand something, you can also mention about that topic in the chat. I will discuss in a